Yeah, so I was just not saying much. I was just saying that thank you. Thanks a lot for the introduction. Yeah, so what I'll do is to give a bit of a background first on psychedelics themselves. And then I'll show a series of some findings from my group and some of the data set that we have collected and so on. So yeah, okay. So I will focus on how the psychedelics act in the brain. So that's the main focus. I'll try to keep it simple in the beginning, at least, before we go into a bit more advanced stuff. Okay, so a bit about what we are doing in my lab. As you were I was mentioning in the introduction, I'm an electrical engineer. So, and my bachelor's, master's, PhD, everything. I was doing something else like building wireless systems, absolutely different from what I do. And then I had a career change and started, working in neuroscience, where I used my quantitative background in engineering and statistics and maths to understand how the brain functions. So that's the question I'm trying to find answer to, like many others in this area. So for this, I use computational models. So I run a computational neuroscience lab. We build models. We develop methods towards the end to understand what are the mechanisms of neural mechanisms of different brain functions. So how do we do that? So as I said, I am a computational or theoretical neuroscientist. So I build theories. We run experiments to validate, to test our theories, as well as we also develop work with clinicians to develop better treatments. How do we do that? So in terms of theory, we develop Bayesian framework or machine learning or statistical frameworks to infer changes in brain connectivity, how different parts of the brain talk to each other using non-invasive brain imaging techniques like MRI and EEG and MEG and so on. And we use interventions. For example, today I will discuss psychedelics as a way to, as an intervention. So there are 3 different lines of research in my lab. So as I said, we develop methods. So that's the first thing. Towards that end, we have developed a sophisticated framework. This is what we call dynamic causal modeling, which is to infer connectivity changes in the brain using non-invasive brain imaging methods like MRI and ETC that I was discussing before. Secondly, we use psychedelics, which I'll talk about today in detail. And the third one is that all the understanding that we generate about how the brain implements different things like learning and all that, we try to build better AI using that understanding, but that's a different topic. I'm not going to touch on this. I'll touch mainly on second, using some of the methods that we redevelop in the first point. Okay, so let's start. So I use I'm an electrical engineer, so I try to understand things by breaking them. So my interest is in consciousness, and of course, in order to understand the brain, while you have a few neurosurgeons here who actually literally break the brain to treat it, I use other ways, which is to use psychedelics to alter or change the consciousness or the brain states. And once I change, I induce this change, I measure this using using non-invasive brain imaging. So my interest in psychedelics is as a way to understand consciousness. That's the primary focus. However, of course, many of you know that psychedelics hold huge potential to treat many mental disorders like depression and anxiety and so on. So this is huge, very, very topical these days. Lots of work being done in this area around therapeutic and clinical research and using psychedelics. One thing which I will touch upon during the talk, especially when I talk about a data set, a new study that we have recently finished, is about what we call set and setting. So set and setting is about the context. So in what context, in what setting, in which environment, and how do you prepare your patients to give them this treatment? And this is extremely important. So which requires a lot of discussion around traits and dosages and environmental factors and pre-states and how you approach the session, the administration session of the drug and so on. Sorry. Okay, so I will touch upon that as well. However, my main interest, as I said, is in understanding neural mechanisms of consciousness by understanding altered states of consciousness. And I'll talk about that. How do I actually do this? Okay. Yeah, so first of all, I'll just start with some basics on subjective effects. So before that, I would just talk a bit. So when I show this slide, I give, first of all, I qualify that here that this is not about, so all the results on this slide are where the psychedelics which are here at the very end of this spectrum, they are under a very controlled environment. So what I'm showing you here is this, that these psychedelics, their harm to others and harm to users is actually extremely low. Yeah, compared to lots of things like cocaine and heroin and even tobacco, which is widely used, and alcohol in some of the societies. You could see that LSD and magic mushrooms, which is basically psilocybin, they are white here. But as I said, this is the qualification is this, that when these drugs are taken under a proper monitoring and with a therapist and all, right? So it's not like you just take it wherever you want on the street or whatever. Yeah. Okay. So this is one thing. So this is an extremely eye-opening data point. So the harm to others and harm to potential and harm to yourself is extremely low with psychedelics. Secondly, we'll talk about the subjective effects, like what actually psychedelics induce, right? So there is something what we call a very well defined now and well validated questionnaire. This is what we called 5 dimensions of altered states of consciousness. So this is something here. So we call this 5D ASC because this questionnaire has 5 dimensions, and these dimensions are oceanic boundlessness, OB. So this is about experience of being oneness with the universe and ego dissolution and all that. Then AED, which is the anxious ego dissolution. Then VR, which is one of the most you could say most salient feature of psychedelics is this, that people have visionary restructuralization, which is about having visual hallucinations and seeing very vivid colors and all that. Then there are auditory alterations, and then there's a vigilance state as well. So these are the main 5 dimensions in which we measure the subjective effects of a psychedelic drug. Here you see this is a meta-analysis. These white blocks are the placebo, and this is for the LSD. LSD is a synthetic psychedelic compound, and these black bars, these are like with psychedelics. So you could see that, for example, oceanic boundlessness, which is one of the most pronounced effect, and also visual hallucinations, they are pretty high. Each of these scales are subdivided into subscales. So, for example, OB, oceanic boundlessness, the subscales, for example, experience of unity, spiritual experiences, blissful state. So people report a lot of this in our experiments and others. So which you see is pretty high here. Then anxious state, anxiety. So this is pretty low. So I will talk about some of the work that we have done, and in that we see that like people may have anxiety attacks or panic attacks, but it's very rare. And then also visual hallucinations are pretty high as well. Similarly for psilocybin. So psilocybin is another psychedelic compound, which basically, which has magic mushrooms as the active component. So LSD is synthetic. This is more natural. So same data, different spider plot. Again, these different subjective effects like this blissful state, and anxiety is pretty low. These 3 colors, they show like a red one shows low dose of psilocybin, green dose shows moderate, and the brown or whatever color is that, the dark color is the high dose or the full dose. Full dose of psilocybin means 25 milligram, mid dose around 18 to 20 milligrams, and low dose around 10 to 15 milligrams for a usual body weight. So what we see is a very similar kind of we see that lots of blissful state, even with a moderate dose, you can induce these subjective effects here. Okay, so that's a bit on the subjective effects of psychedelics. Then I would like to talk about a bit of a theoretical foundation. Some of the so psychedelics as a unified therapeutic theory. So what is this? So there's a theoretical work about how the brain functions, and this is what we call hierarchical predictive processing. This is a theory which talks about how the brain functions, which says that brain is actually hierarchical. So I show this as a pyramid where there's a sensory information which is entering here, and then at top is the high level inferences. There are bottom up prediction errors, and there are top down predictions. So basically what the brain is doing, according to this theory, brain constructs the top down explanations for the bottom up sensory signal. And because, as I said, this is a brain as a hierarchical organ. At every level of hierarchy, there is this, you see these hierarchies here, where these are canonical, what we call canonical microcircuits, which are, which are shown here. So there is this prediction error going up, or the sensory information moving up, and the predictions moving top down. And what does it mean? For example, you could say that subcortical areas like thalamus, which controls the information flow to the cortex. For example, I'll show this example, some empirical work on in a couple of slides on this. So subcortical areas, you could think of limbic areas, and then cortex. This is where the high level inferences are happening. The self resides there in a sense. So this is this is a view about how the brain as a hierarchical predictive processing organ. And now, what the psychedelics do? So psychedelics, basically, we say that this flattens the hierarchy. So they collapses the hierarchy in a sense. So the idea here is this, that the brain, the higher level areas in the brain, like in cortex, they are creating these, they are producing these predictions about the world, that how the world is outside. Yeah, so these are the top level signals which we are generating. And then the idea here is this, that in many of those internalizing disorders like depression and anxiety, these predictions, in a sense, they become stuck there. Yeah, so this is an example of very rigid beliefs about the world. Yeah, so you are in depression, for example, you have a very rigid beliefs about a state of the affairs around you. Yeah, so what the psychedelics do, they reduce the precision of the top-down prediction. So what they do is this, that they introduce uncertainty in the beliefs about the world, in a sense. And they also, in a sense, disable the or suppress the bottom of prediction errors as well. This is what hypothesized as how the psychedelics and at a systems level work. So what the second, by flattening this hierarchy, what we are saying is this, that a psychedelic relaxes the beliefs in the higher level of the hierarchy. So there are relaxed beliefs about or the rigid beliefs which person has about the world, they're relaxed. Yeah, this gives people opportunity to explore landscapes of alternate hypotheses or new way of thinking. So that's a theoretic framework. This is which comes from predictive coding or hierarchical predictive coding processing framework, and combines this with psychedelics and how they actually could help people with some internalizing disorders. Now, so this is the working, in a sense, a theoretical account of how psychedelics treat mental illnesses. I will show some examples of this work as well. So what I'll do next, the next slide has a bit of modeling work on this, and this is where our modeling framework or dynamic causal modeling enters. So this is the only slide where I have some equations and some models. This is not to in a sense harass you. It is just to show that there's a modeling framework that we have developed where you can actually develop a generative model of how to explain the measurements or the non-invasive measurements, like using MRI or EEG. You have, for example, you have observed data. This is what we call Y, and then we want to actually estimate what might be happening inside the brain, at the source level. So you have a sensor level data, and you want to look at the source level neural interactions. These are they are shown by theta. So this is basically what we call the effective connectivity or the directed connectivity between brain regions, and this is like what we want to infer using our MRI or EEG or MEG data. For this, we have developed these framework where you can look at whole brain, and you can look at system level or macro scale connectivity between brain networks and so on. You can look at even excitatory and inhibitory influences and things like these. This is the framework that we have developed just to introduce you, because I will show the application of these methods. So I just had this slide here. So So here there's a first study which we published a few years ago. Here we test the hypothesis that I was showing before about hierarchical predictive coding or hierarchical predictive processing. So there's something what we call a thalamic filter model. So thalamic filter model is a very influential model coming from animal studies, which says that thalamus, which is a subcortical area in the brain, it works as a filter. So all the sensory information, visual and auditory and all that, it comes to the thalamus, and from there, the thalamus is going to control how much of the sensory information is to be routed to the cortex. So it works as a filter. Yeah, there's lots of information which is coming through all our senses, and thalamus is controlling the information flow to the higher order regions in the brain. So this is an example where we are showing that there is this posterior cingulate cortex, which is basically a cortical area, higher order cortical area. And what we are showing here is this, that under the influence of LSD, there's a lot more information that enters through the thalamus to the cortex. So under psychedelics, the idea is this, that this filter opens up, and there's lots of information which flows to the brain compared to under normal physiological conditions. Under psychedelics, this thalamus is like has high connectivity to all the brain areas and so on. So we published this paper a few years ago at PNAS, which showed the first in vivo evidence of thalamic filter model in humans. So here we have so these are people who have been given LSD, and in this we also give a pre-treatment with another compound called ketenserine, which is a 5-HT2A antagonist. So basically what we are showing, I don't want to go into the details, but using the methods that I showed you in the previous slide, the dynamic causal modeling, what we showed is this, that there's under the drug versus placebo condition, we see there's an increased connectivity from thalamus to the ventral striatum, and there's a increased connectivity from for example, from PCC to ventral striatum and so on. While what we show is this, that under the blockage condition, where we do the pre-treatment with ketenserine after and then we give LSD, we see that we don't we see the opposite effect. So in that sense, this is an evidence that there's a lot more connectivity from thalamus to the cortical areas, and provide a bit of a very first paper that provided evidence that this might be the mechanism by which psychedelics work in the brain. And now there are several more papers have appeared, not only in LSD, which we used here, but across other psychedelic compounds like psilocybin and MDMA and DMT and so on. So people have shown very similar. This has been replicated many times now by several other groups as well. So that's one of the work. So this led to a working hypothesis of how psychedelic-induced changes in brain connectivity. So this is basically where what we call the cortico-striato-thalamocortical loop. So here, for example, there's a thalamus, and then in these this blue color areas, they represent the association areas, while the purple color, they are the sensory cortices, what we are showing is this, that the psychedelics increase the connectivity between the thalamus and the sensory areas. So for example, here, as you see, or here, and decrease the connectivity between thalamus and the association areas, for example, here, or as I showed in the previous slide, there's an increased connectivity from thalamus to the PCC, for example. So this means that what's happening is this, that there's a reduction of thalamic filtering, as I showed you, which results in increased connectivity between sensory areas and the rest of the brain, and there's a decreased connectivity between association areas and the rest of the brain. So this disbalance between the sensory processing and the integrative processing results in the psychedelic experience. So this is a hypothesis, working hypothesis, for which several studies have shown some evidence. So next, I would just quickly want to show a couple of other studies. So this is a study another few years ago. This is without psychedelics. This is the data from human, not from the Human Connectome project, but from another large data set of around 400 people, where we looked at 3 different large-scale vesting state networks, like default mode network, salience network, and the dosal attention network. So the default mode network, I guess some of you might know, this is one of the most studied and most popular kind of a network, which is about self-processing. So this is like when we are thinking about ourselves, basically default mode network in a sense is activated. Dosal attention network, on the other hand, is about the external world. It's about the object. So this is about external, when we are attending things. So this is the network which is activated when, and then the switching between the self network and the outside network is done through salience network. So what I'm showing you here is a functional connectivity matrix between. So these are the nodes of or regions in a default mode network, salience network, and dosal attention network. And what I'm showing you is a replication of a very seminal result that shows that these networks, the DMN and the 2 other networks, they're anti-correlated. So this is a very seminal work from Waco, at University of Washington, St. Louis, possible candidate for a Nobel Prize even. So because these networks and these anti-correlation have been implicated across many diseases like Alzheimer's and depression and so on. So this is, is it, Athar, you want to say something?
Yeah. First of all, is it okay, Razi, if I interject?
Yeah. Why not?
Yeah.
Why not?
So how did you do this correlation?
This is basically, what you do is that you have these regions of interest, for example, PCC and middle prefrontal cortex, and so you can use some atlas, and then what you do is that you extract the signal from these brain regions and then calculate correlations. So what I'm showing you here is that default mode the areas in the default mode network, they're anti-correlated. So you see these blue areas, they are anti-correlation. So these are anti-correlated. So the default mode network is anti-correlated with the other, what we call the task positive networks, like dosal attention network and salience network. So this is a widely replicated, widely celebrated result that these networks are there, and they are, basically what you do is basically extract signal from these regions of the brain and then calculate correlation between them. Yeah, and then show them as a connectivity matrix. So however, I mean, the interesting thing here is that this is only correlation, and they don't tell you the causation. They don't tell you what brain region is actually influencing the other brain region. It just shows you that these are correlated, but they don't tell you that what is the causation, which is which brain is it the default mode network which is influencing salience network, or is it other way around? And this is important that we need to know the causation in these networks, because some of the hypotheses that I was talking about, that thalamus has increased connectivity to the cortex. So clearly there's a direction involved in it. We need to know that which area is influencing other area. So for this we have something like this now. So this is using dynamic causal modeling, where what I'm showing you here is the same brain regions What we are showing here is this, that first of all, these networks, they are grouped together. So this is, default mode, salience network, total attention network. Then what we are saying here is this, that the task positive networks like the salience and dorsal attention network, they inhibit default mode network, while the default mode network excites task positive network. So this is like what we're trying to show is that there's just not correlation, but there is actually underlying excitatory and inhibitory influences in a certain direction, and they have these valences, and I show this on the brain as well. So this is kind of, again, a seminal kind of a paper now that we published in 2018. Now, this is all without psychedelics. Now, the idea here is this, that we wanted to see if this anti-correlation pattern changes under psychedelics. Yeah. So here comes the idea. So one of the main subjective effects of psychedelics is that people report ego dissolution. So ego dissolution is a sort of a experience where people say that their internal world and the external world is basically in unity. So there's a blurring of boundaries between the inside and the outside, or the blurring of boundaries between the object and subjects, So so this is what we wanted to see, that how this relates with these networks, right? So as I said, default mode network is about internal processing, is about the internal world, while the salience and the dorsal attention network, they are about the external processing. And these are anti-correlated, as I showed you before. So we wanted to see that when people are reporting that they have ego dissolution, what's happening to these network and their anti-correlated structure. So what we are showing here is a placebo effect, where we have these networks, three networks. Default mode networks, sometimes people also relate this to narrative self. So and salience network, people relate this to minimal self as well. So these are some constructs about self. So in this experiment, what we have is a placebo, and then we give these people 100 milligram of LSD. And then after about 75 minutes, we again do an MRI, resting state MRI scan, and then calculate changes in brain connectivity. And then we do another brain scan after 300 minutes of LSD ingestion. Yeah. What we are showing here is this, that there is a, after 75 minutes of administration, this connection between between DMN to DAN, which was a inhibitory connection, becomes an excitatory connection. Yeah, and similarly, this connection, which was an inhibitory connection from salience network to the DMN, becomes an excitatory connection. So there's this this correlation structure is going away in a sense. And then as the LSD, it has about a half-life of about 4 to 5 hours. Once it washes out, kind of people come back to the normal state. So what we are showing here, without going into too much into the data, what we are showing is this, that under LSD, people have the anti-correlation structure between the networks, which are about the internal about the internal self and the external world. The anti-correlation structure actually blurs, as the people experience ego dissolution. So that's probably one of the neural mechanism that might be at play. Then we are doing some regional analysis, not necessary to go into this. So I think I'm getting a bit close to 40 minutes. So I will try to finish with. Now I have a few more slides, so maybe I'll need another 10 minutes. So this is where I would like to introduce a study, a clinical trial in my lab that we finished. So this is what we call PsyConnect. So this is Australia's first and world's largest study where we gave psilocybin to 60 healthy adults. So this is, while we call it clinical trial, it's about healthy population. There's no clinical population in this. What we do in this trial is this that we have two groups of 30 people each, and one group does a meditation course. So in the beginning, I was talking about set and setting, that how important it is, like the mindset and the environment and the contextual factors in the psychedelic experience. So we hypothesize that that meditation, a short meditation course, will help people with their psychedelic experience and will result in changes in brain connectivity, which will be conducive to better health outcomes. So what we did is, as I said, 60 people, half of them did a short 8 weeks meditation course. We also use music and movie watching. This is what we call naturalistic stimulus or naturalistic imaging. It was a multimodal trial in which we did both functional and diffusion imaging, as well as EEG recordings were done as well. So we did imaging at the baseline and then on the day of drug administration. It was finished last year in December. It's almost an year. We have some longitudinal follow-up with survey questions and all that. So we are finishing this this week. So this is this trial is now completed. We are hoping to publish these results. Now it's already late 2023, but we are hoping to publish these some of these early results in the next couple of months. A bit more about the design. So it's because this is here we are not using any clinical population, we are just giving it to healthy people. So we have a very tight screening protocol where people apply. So although we had only 60 people that we wanted to recruit, we had more than 3,000 people applying to be in the trial. So we had to do a huge amount of screening of people using various different psychopathology screeners. Then half of them do a meditation course, which was administered by the Oxford Mindfulness Center. It's a usual what we call the meditation-based cognitive training test, MBCT, 8 weeks of MBCT course. Then we do imaging, an MRI and EEG at the baseline without drug. During the MRI session, we collect resting state data. We collect data when people meditate while lying in the scanner, while they're listening to music, while they're looking at a movie and things like that. And then after a week, we give them psilocybin, about 19-20 milligram of psilocybin, and then we repeat the same procedure of imaging, which is basically same sequences as the baseline scan, resting state scan, and meditation scan, music and movie watching. Then some very preliminary results from this trial compared to some behavioral results compared to baseline. Psilocybin greatly increased liking of ethereal music across both groups. So without drug on the baseline, when people were listening to the music piece, many people didn't like it. They found it boring or something like that. But under psilocybin, people reported that they loved the music. There's lots of work on that there's a synchrony between music and the psychedelic experience. Psychedelic experience basically enhances with the music and people get more out of it. We also see that psilocybin subjective effects, they are meshed evenly between both meditation and non-meditators groups. So as I said, there are two groups. One do meditation of 8 weeks, other don't do, but we see that subjective effects are pretty evenly meshed. A bit of results used with imaging. We looked at, it has taken a lot of time. The whole year we have been analyzing data. So this is very preliminary and possibly hopefully in the first paper, we will have a bit more results which are more at whole brain level rather than region specific, but some initial models where we look at egocentric self-referential brain regions which relate to bodily self and visual processing. So mainly what we see is for example, in the resting state scan, we see more top-down connectivity mostly, and then when we look at DMN and visual areas, we see very similar patterns as well. These are just very, very preliminary results. When we look at meditators versus non-meditators, what we see is this meditation showed there's a amplification of changes in brain connectivity in the default mode network compared to the non-meditators. And also we see that people who meditation trained participants, they also showed increased subjective experience of disembodiment. So lots of people actually reported that they experienced disembodiment or out of body experiences and things like these. So we are looking at the neural mechanisms of those things as well. I think it's the last slide, I think. So some of the conclusion is this, that the context is important, like music and meditation. They modulate the effect of psychedelics. Psilocybin increased region sensitivity to inputs, and some of the ongoing work that we are doing, we are measuring modulatory changes and looking at neural mechanisms of disembodiment and so on. So these are all very preliminary. Results will come out, and we will do a preprint in a few months, and then we can discuss maybe in a different session what the actual results are, just to give a bit of a taste what we are doing and what the study is about. So with that, I would like to thank everyone. Lots of people in the lab, my collaborators, many of the funders, and thanks to all of them. And thank you, and happy to